dance by the water neath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me? By the water neath the Mexican sky. Drink some margaritas by a string of blue lights.
Yeah, Detlef told me that I had to introduce him in 10 seconds. The guy has 59 pages of CV, and he wanted me to introduce him in 10 seconds. So okay, I'm going to do my best. So one of the things that is very interesting about Detlef is that he made his PhD in genetics in Drosophila. And then he moved to plants. So it's one of these people that select plants because he loved plants, I guess. I don't know why. So he did his postdoc later in the US with Elliot Meyerovich, that probably all of you know. So he helped put in a lot of letters in the ABC model back then in flowering development. And then he was a PI in the Salk Institute for quite a bit. And then in 2001, he became a Max Planck director. And now, since then, he is a Max Planck director in the Max Planck of Developmental Biology for the old people, and Max Planck of Biology Tübingen now, that you may know. So he received like an impressive list of awards: Barbara McClinton over Nordic, Doctor over Causa in Sweden, the Life Achievement Award last year. He's a member of the National Academy of Science of the US, the German one, the EMBO, the Royal Society, and all the societies and stuff that you can be a member. He produces an amazing number of papers, so you want to know how many? So it is in total, until I got the CV, is 462 papers. From those, 19 are nature, 17 are science, and most impressively, 26 are plant cell. That's even more important. Well done, Detlef. He also trainee 146 people. Many of those, you hear them here. For example, Hernan that came here, Corbidian that was here. Who else? Miguel Blasquez, you all know, Francois Parcy, and of course, the most important one, the golden age of his career is when I was in his lab. And he won't admit it, but it's true. Yeah, so, and as I told you a couple of weeks ago, he's a legend, so you will enjoy it. Thank you for coming, Detlef.
Thank you. Thank you, Pablo. That was very nice, a little bit longer than 10 seconds. Very happy to be at the Instituto de Hortofruticultura Subtropicale Mediterranea, and to speak about my research. So we go here, and let me see. If you help me to get to my presentation. Should be it's open. Yeah, exactly. So, very. No, it somehow doesn't show. So, or do we have to? Do we just launch it, or how do we project it? How do we? Yeah. Very good. Okay. All right. So, you might wonder what I will be speaking about. Plants live in an imperfect world, and so do we. So what I want to speak about is variation, phenotypic variation. And I want to remind you that the ability of organisms to very quickly adapt to their environment is really remarkable. And I think everybody here works on plants, but we shouldn't forget that also animals can do this. So this is actually the same animal that you're looking at. These are two pictures, courtesy of my colleague Gilles Laurent from the MPI for Brain Research. And you see this beautiful cephalopod on different backgrounds. So organisms can adapt very quickly in real time to their environment. But doing this comes somewhat at a cost to do it all the time, to change your phenotype in real time, and that is why organisms also vary genetically, so that they also have already a little bit of a leg up. And here is one particular species with enormous phenotypic variation from the good to the bad. And I'm not going to comment on that gentleman on the right. He did good things in 1981, so we probably should stop there. So you look at these individuals here, and how much of their behavior is genetically encoded, I don't know, but certainly when you look at their phenotype that we see, that is genetically encoded. And this is what we've been interested in for the past 25 years or so, why there is so much genetic variation in the world, and this is what I want to talk about in the next 45 minutes or so. And one answer is, of course, that the environment varies tremendously, but not only does the environment vary today, the current environment, but there is change in the environment, and this is from a paper about a decade ago looking at what's called the perma-drought severity index and how that's going to change at the end of the century versus now. And one of the things is actually really quite interesting. You look here in the center of Europe, you have some of the largest changes compared to, say, the Sahara. So in the Sahara, it's already extremely dry, so not much can change, but the center of Europe is really very much a threat of changing climate. So environment is going to change, and obviously we weren't the first ones to think about how plants going to deal with the changing environment. This is something that ecologists have thought about for a long time. And the conventional wisdom is that species adapt by just migrating to the places that they already adapted today. So if you live here further in the south, then what the textbooks will say is you just move to the north, or if you live lower in the mountains, you just move up the mountains. And this is just one of many, many papers that show this. That, so how this species here continues to migrate northward. And this is a plant. So this has a couple of problems. Plants obviously can't walk, but they can migrate with seeds. But from an ecologist point of view, a more serious problem is that this scenario relies on the populations that are at the edges of the entire population. And these populations here are fragmented, they have low genetic diversity, they're actually the least adapted populations, and they are supposedly the ones that will rescue a species from their demise. So one of the things that we've been very interested in is figuring out how organisms deal with the variation that they face today, and then from that to deduce how will they do in future. And so we've been working with Arabidopsis thayana. I apologize, the PDFs somehow didn't quite make the transfer from the Mac to the PC, but I think you can still see this. So we've been working with Arabidopsis thayana, and we've really, together with our colleagues, have been developing it as a model for ecology and evolution over the last 25 years. And this just shows strains of Arabidopsis from all over the world where we have genetic information. Colored are the ones that were in this collection of over 1,000 strains where we published genetic information about a decade ago. Like humans, Arabidopsis actually comes out of Africa. This is where you have most genetic variation. Today it's restricted to the mountains in Africa. It then populated Europe, and from there spread to Asia. And then much more recently, it was brought to North America, was brought to South America. So we've actually worked on Arabidopsis Patagonia together with Javier Botto, and then also here to Australia and New Zealand. And this is just some photos that show you the impressive variety of where Arabidopsis grows. So here near Tübingen, in abandoned train track, oak forest in Spain. This is Patagonia. Here, this is not a staged picture on the Baltic Sea, and here high up on the mountain in Africa, Kilimanjaro. So very different places that Arabidopsis grows, and that already tells you that they must be adapted to different places. Now, the way that ecologists typically look at this, that they do this reciprocal transplants, where they take a strain or a population from one place, and then they grow it somewhere else, and it's called a reciprocal transplant because you do the same for this other population and transplant it. And then on average, you have a home advantage. So as Pablo mentioned, I was for many years in Southern California. So if the Padres play at Petco Park in San Diego, they tend to win, whereas when they play in LA in the Coliseum, they tend to lose. And this is how it works when you transplant plants, the typical result. But this is one strain at a time, so how do you generalize from this to predict what happens in future? So I was extremely fortunate when a Spaniard from southern Spain, from Alicante, not very far from here, came to me a little bit over a decade ago, and suggested an experiment that I thought was completely insane. I thought there's just no way that a young graduate student could pull off such an experiment. And to be honest, I didn't fully understand what he wanted to achieve with this experiment. I said, you seem to be very enthusiastic, so just go ahead and do this, Moy. So what Moy did was he took hundreds of strains of Arabidopsis thayana from different places in the world where we knew the entire genome, and then he grew them either in Tübingen or near Madrid, and he used these rainout shelters so that he could control how much water these plants would get. And then he simulated both a rainfall regimen that you would have in Tübingen and a rainfall regimen that you would have in Madrid. And obviously in Tübingen it rains a little bit more than it does in Madrid. And so not only is Moy extremely enthusiastic, he is also very charismatic, and so he convinced the entire lab to travel to Madrid in winter to look after the plants. And I wonder why people agreed to that, but somehow they did. All right. So looking at these plants, you won't be surprised that rain is good for plants. You see these plants do a lot better than here where plants are exposed to a mild drought. So the sort of rainfall regimen that you would have in Madrid compared to the one here in Tübingen. And so thousands of pots imaged over many time points, so hundreds of thousands of images, and then counting survival of the plants and also the seeds that every plant produced. And growing plants either as individuals where there's one plant in a pot, or these populations where there are 20 plants in a pot. And this is then the result from this massive endeavor, looking at Madrid and Tübingen. First thing that you see is that rain is good, so you produce more seeds when there is rain compared to where is drought. And if there's ample rainfall, then it's better to be somewhere where it's warm, so you make more seeds than somewhere where it's cool. Here in Tübingen, you make fewer seeds, but under drought, this goes the other way. Under drought, it's better to be somewhere where it's cool because the water doesn't evaporate as quickly, so you can still make some seeds, whereas in Madrid, under drought, most plants actually die and don't produce any seeds. And I should mention the number of seeds. That's important because it's a proxy for fitness, Darwinian fitness. Strictly speaking, it's not just the offspring that you have, but strictly speaking, it's actually the number of fertile offspring. So you would have to go to the grandchildren, which we didn't do. We just went to the children and counted those. And then it also makes a difference here. High density, you produce more seeds than if it's just a single individual. Fine, this is just the raw data, but what can we actually do with this? So the charm actually came from the fact that we had genome information. So not only can we ask, is a strain that comes from the north of Sweden, how does it do in Tübingen versus Madrid? Well, we can go along the genome, and we have millions of SNPs. In every place in the genome, we can ask if you have one allele versus another allele, what is better? So Arabidopsis thaliana is typically inbred, so we have the same allele, large A at whatever point in the genome it is, versus small a, and we can ask all the strains that have the large A allele versus all the strains that have the small A allele, who does better? Or is there even a difference? And it turns out that differences are again massive. And we can then ask whether it makes a difference where an allele comes from. So this is shown in this diagram. We have a map, we know where all these strains that we tested come from, and then for every allele, say we have an ochre allele and a purple allele, we can ask where do we find it and which strain. We can draw the distribution, and then we look at the centroid of the distribution, and then we define this as the average climate where an allele comes from, the purple allele versus the ochre allele. So we look up all kinds of climate factors for these different places. And this is then the outcome of this experiment after doing all this work that I just described. And two axes of climate variation that are important for Arabidopsis thaliana is the seasonality of precipitation. So seasonality here is very high, because in the south of Spain it doesn't it normally doesn't rain in summer, but it rains in the winter. So high seasonality, whereas in Germany it rains more evenly, so low seasonality. And then the total amount of annual precipitation, which is low here and higher in Germany. And we're looking at millions of alleles here, so you see right away, if it is an allele that comes from a climate that's similar to Madrid, then that's positively selected on. That's shown here in green to dark green to light green. So these alleles are positively selected in Madrid, meaning they contribute to survival. Versus here, alleles that come from a climate that's more similar to what we have in Tübingen, they not only are neutral, they are actually being selected against. So you see red indicating selection against. So if you have an allele from one of these places, then that is not only just, it's not that it doesn't matter, it's harmful. So home alleles on average are good, and the away alleles on average are bad. And this is what evolutionary biologists call antagonistic selection, because if you think about this for a second, the best situation would be if you have alleles in your genome that are good, say, when you are in Madrid, but are irrelevant when you are in Tübingen. But here, what we find is actually there's this cost. You have an allele that's good in Madrid, it comes at a cost if you have this allele when you're in Tübingen. And we can think about this a little bit about humans. So if you transplant somebody from southern Spain to Tübingen, there probably is a cost that in winter they're not quite as well adapted and don't do as well as somebody who comes from the north of Sweden and is in Tübingen in winter, right? So what we learn here is the world is complex. And so I already gave you an answer. This is a rhetorical question here. Selection also should be complex. That they are all these antagonistic forces. And that's really what makes studying evolution so interesting, that the world changes all the time. And because the world changes all the time, therefore these selection regimes change all the time. And so here's an long dead white man from England who put it in a more lyrical way: one man's poison, another's meat. Or more recently, one of the most important philosophers of contemporary times, Yogi Berra, whom some of you hopefully have heard of: if the world were perfect, it wouldn't be. Now, this is somewhat of a joke, but there's something quite seriously behind this. So if you are plant, there are different strategies for adapting to a drought. You can, and this is how ecologists actually call this. The one strategy is the live fast, die young strategy, and the other strategy is this slow and steady strategy. So live fast and die young. You flower very fast. This is an escape strategy. You flower very fast, make a few seeds, but because you flower so fast, there's not many seeds, but at least you make some. Or slow and steady, where basically you grow very slowly. You try to not lose too much water, and then you hope eventually you can, make a good amount of seeds. But what we see right away in Arabidopsis thayana, that flowering quickly normally goes with low water use efficiency. So again, think about this drought escape. You flower very fast, fine, but when you do this in Arabidopsis, you normally don't use water efficiently. So there's more transpiration than if you would flower late. So there are actually two behaviors that go together, but the one behavior, the flowering fast, is good for drought escape. The other one, losing water quickly, obviously isn't good for drought escape. And so when we look here, precipitation warmest quarter, we see when precipitation is low, plants actually tend to flower late and not flower early in Arabidopsis thayana. So Arabidopsis seems to use late flowering as a drought avoidance strategy more than early flowering. And so genetic conflicts, they are common all over the place. They are also common in humans. Probably no human geneticist in the audience here, but I always like to remind human geneticists when we're talking about human genetics that the focus is very much on disease and variants that are bad for whatever disease you're looking at, but you often not considered that there might have been positive selection for certain alleles. And so this is one of my favorite papers from the stack of paper that I just showed you, Huntington chorea. So Huntington chorea was actually the first human genetic disease to be mapped. And the reason, and so it's a deadly disease, so it normally manifests itself when you're in your 40s and you inevitably die, and it's a dominant disease. But it was the first human disease to be genetically mapped already in the 70s, and the reason it could be mapped relatively easily, the end game cloning it turned out to be difficult because it's in a difficult region of the genome, but the reason it could be mapped so easily was because people with Huntington chorea have 2 to 3 times as many children on average than other people. And so I think this is, I'm not sure whether this was from Colombia or Argentina, somewhere in South America where you have these large pedigrees. So it's something that kills you in your 40s, but before you die from the disease, you actually leave a lot of offspring. So again, so it really is in the eye of the beholder whether this variant is actually good or bad. And so this is all to say that context really matters in genetics, and this is why evolutionary and ecological geneticists have come up with all these different terms for these different phenomena that speak to context. So the one that probably most of you have thought about and have accounted is epistasis and redundancy, where it depends on the genetic background whether an allele has an effect or not. So redundancy, for example, often you need to knock out more than one gene, so you knock out one gene, you don't see anything, but then this gene being knocked out when the other gene is already knocked out, you all of a sudden see a phenotype. I just told you about antagonistic selection. There is balancing selection, where it depends often on your neighbors whether an allele is good or not. So this is if you are resistant against pathogen, if you are like everybody else, and the pathogen has a lot of incentive to overcome that resistance. But if you're very different from everybody else, then the pathogen might not care about you because you're really rare. There's antagonistic pleiotropy, so this flowering and water use efficiency is an example of antagonistic pleiotropy, where a single allele affects two different traits, but affects these traits in a way that actually go in opposite direction for whatever is of interest here. And then probably my favorite here, eco-evolutionary feedback, where actually you have an allele, and that allele leads to a change in the environment. Whoever has that allele modifies the environment, and eventually this leads to a different ecosystem structure. And so I started out with humans, and we can perhaps think about how human genetics actually is leading us to destroy our environment. Is it really in our genetics? Are we hardwired to destroy our environment? All right, so give you another nice example of how genetic context matters. This is something that we worked on for quite a long time. It began with work by Janne Lempe and Kirsten Bomblies. Kirsten is now a professor at ETH and was really a chance observation. Janne was working on strains from Umkirch, a little village not too far from Tübingen, and both of these strains look quite normal, but they flower at different times, and that was the reason why Janne crossed them together. And then she found that the F1 hybrids were very sick. And the prominent symptom these hybrids had was necrosis. Now, at first we were scratching our head because we were developmental biologists and we didn't quite know what to make from it, but then we remembered that necrosis is a prominent sign of an immune response in plants. And when we looked at this hybrid and a couple of other hybrids that showed a similar phenotype where the F1s were very small, we saw that the only genes that were changed in their expression, and this is data from the times of microarrays, so really old school, if you will. The only genes that were consistently changed were genes that were related to immune response. So together with the necrosis, this told us that autoimmunity was apparently the culprit, that these plants didn't look so good. And it turns out that we had discovered actually a phenomenon that was well known to breeders and naturalists. There were hundreds of cases. The first case actually described already in 1924 in the literature in both crops and wild species, within and between species, and often simple genetic architecture. Interestingly, although there were many, many papers, this connection to immunity in many of these papers hadn't been made because the researchers sort of had like a mental block that for them it was immunity was only present when there was a pathogen, and they were really confused because these plants looked like they had been attacked by pathogen even though pathogen was not around. And they didn't want to consider that this was genetics. And a friend and colleague of mine, Ginny Walbert, shared with me a number of years ago that when she started as an assistant professor in Stanford, was either the late 70s or early 80s, she actually wanted to make her career working on this, but she was told by reviewers that it couldn't be that plants have immune symptoms without a pathogen, so she gave up studying this. So A few years later, we were joined by Eun-Young Choi, who is a professor in Oxford now, and Eun-Young said, let's just look at this. Is there, well, what is behind this? So we have a few cases, but she really wanted to know whether there was anything systematic about this, how hybrid necrosis arose. And so we became Arabidopsis breeders, made thousands of crosses, and found, yeah, it's widespread, and it doesn't really matter whether plants come from the same place or not. And because we had genetic information, we could relatively quickly map the underlying gene. So these are the 5 chromosomes of Arabidopsis thaliana, and then in color is indicated the density of genes that encode NLR immune receptors, nucleotide binding site loose and rich repeat receptors. And I'm sure you all know that we have, in the meantime, a lot of structural information on this important class of immune receptors, starting with the work from Ji Chai. We call these genes dangerous mix for obvious reasons. Map many of these genes, and you see that often they map to where you have a lot of NLR immune receptors. Indeed, we show that the NLR immune receptors are causal. We have different types of genetic architectures. So here's one of our favorite ones, DM2, produces all these different risk alleles that interact with with alleles throughout the genome. And then this is also a really cool case, RPW8 and RPP7. There are 3 alleles at this locus, 3 alleles at this locus, and they interact in an allele-specific fashion. Now, it shouldn't have been a surprise to us that NLR genes came out as the top culprit, because we already knew from earlier work that had been led by Richard Clark, who's now at the University of Utah, that NLR immune genes are the most variable genes in the plant genome. So that sort of makes sense. What gives you trouble when different strains get crossed to each other? The genes that are most variable. And so the one way to think about this, you bring together genes that have not seen each other, because you can't test all these different combinations. You see genes that haven't seen each other, and therefore bad things basically happen. And so we then went on to study the molecular mechanism. I have to admit, I, as a geneticist, would have been perfectly happy to stop there, but I'm friends with people like Jonathan Jones and Jeff Dangle and Brian Staskowitz, and they tend to be bullies, and they all say, Weigel, you gotta find the mechanism. This is not good enough if you just study the genetics. So that's what we did. So this is a case that Zip Tran looked at, where both sides, dangerous mix 1 and dangerous mix 2, both of them encode NLR immune receptors. And so with help from the lab of Jeff Dangle, we were able to figure out that they can actually form homomultimers, both DM1 and DM2 alone, but they do not signal as homomultimers. So the one parent makes DM1, the other parent makes DM2D, and then they are perfectly fine because nothing happens. But then in the hybrid, DM1 and DM2D monomers come together. They also oligomerize, but because they are not properly adapted, they are apparently in a conformation that now signals, and therefore signals danger, presence of pathogen when there are no pathogen around, and that's not good for the plant. And then this other case is one that Lei Li, who leads his own lab at the Chinese Academy of Science now, together with Annette Habring, figured out, where it's an interaction between an NLR immune receptor, RPP7B, and another immune protein called HR4. So the standard version of HR4 is a protein. It's a small protein that is being used by the plant to defend itself against pathogens. It goes to the membrane, makes holes in the membrane, and that's part of the immune response. And so this helps the plant to defend itself against pathogen. What we hypothesize, we haven't seen this directly, is that, but this fits with how we think about NLR immune receptors and effectors and so on, that a pathogen, to suppress this response, will inject a protein into the plant cell called an effector protein, and this effector protein will modify this HR4 protein. So it's now modified. It cannot go to the membrane anymore, and therefore the immune response is suppressed. But this is where the NLR immune receptor comes in. It recognizes the modified HR4, and recognizing it leads to formation of this oligomer, and it now signals danger, and immune response is mounted. And what happens in this hybrid is we have a different version of HR4. It's structurally different, so we showed that it's structurally different, and this is sufficient in the absence of a pathogen to form a multimer, which again we showed biochemically. So two different ways of how NLR immune receptors through inappropriate resistosome formation can lead to autoimmunity. And I forgot to mention these complexes are generally called resistosome in analogy to the inflammasomes being formed by mammalian NLR proteins. So how did that help us to think about disease resistance in plants? If you go to a field, many microbes, some of which are pathogens. If you look at an individual plant, you'll find an individual plant can resist a few of these because it has a certain spectrum of NLR immune receptors encoded in its genome. If we go to all the microbes in the field then and all the plants, we will see that individual plants resist a few of these microbes, but together, actually each of these microbes can be resisted by some plant. Now, if in principle, plants can resist every one of these microbes, why don't we have these super plants that can resist everybody? And the answer seems to be is that you have to encode a very large number of NLR immune receptors in your genome, and the chance that something goes awry, that all of a sudden inappropriate resistosome formation happens, goes up obviously exponentially the more alleles you have. So this is verboten because there would be autoimmunity. So there's a natural limit to the number of immune receptors you can encode in your genome. And interestingly, that limit seems to be species specific because in different species of plants, there's a different number of NLR immune receptors. So some have only a couple of dozen. Papaya, for example, Arabidopsis has on the average about 150, and then wheat can have 2,000 or so. So different tolerance, and tolerance is somehow species specific. Now, one of the questions that I always got asked when talking about this, whether there's evidence for anti-hybrid necrosis, so where actually genes come together or proteins come together and you suppress immune activation. And this is a story that began many years ago when we were looking at plant size, and Marco Tedesco noticed that natural strains of Arabidopsis thaliana that are smaller tend to also make leaves more slowly and then often suffer from this early onset necrosis. And you will remember from earlier necrosis indication of autoimmunity. And so Marco, he's now a professor at the University of British Columbia, ran actually, that was the first successful GWAS in plants, and he found that there was a single gene, ACD6, where allelic variation determined whether or not you had this phenotype of making leaves more slowly, being smaller, and having necrosis. And then he showed that indeed, when you knock down this gene, plants become much larger. So these are different wild types, and in each case, you're knocking down ACD6. The specific alleles makes these plants much larger, and the necrosis disappears. So dead cells here, and no more dead cells when you knock it down. So this seemed to be somewhat confusing. Why would so many strains, about 10% of natural strains of Arabidopsis thaliana, have a gene in the genome that makes them much smaller than their neighbors? And again, necrosis linked to immunity, so not very difficult to come up with this hypothesis that the special allele of ACD6 that we first found in the EST-1 strain provided protection against pathogen. And indeed, we first looked at this fungus here, Golonomyces orontii. If we knock down the ACD6 gene, plants become susceptible, where before they were resistant. Not quite as susceptible as the reference strains of Arabidopsis thaliana, but much more susceptible than the EST-1 strain. And that's just one pathogen that will be somewhat boring, but then we, with the help of various colleagues, also looked at oomycetes, we looked at bacteria, we looked at insects. In every case, the strain that had the ACD6 EST allele was much more resistant than the one where the gene was knocked down or absent. So it seems to be typical growth defense trade-off. when you have this allele, that's good in an environment where there are a lot of pathogens around. You stay small, but at least you don't die from pathogen attack and can make still a few seeds, whereas the other allele is better when there are few pathogens, because then hopefully you don't get infected and live to make a lot of seeds. Now, what we had noticed, or what Marco had already noticed, was that the effects, though, were not uniform and that they depended somewhat on genetic background. So yes, a lot of strains that have this ACD6 EST allele show a lot of necrosis, but there are also strains that don't show necrosis. And this is just the genetic evidence. You can take the ACD6 gene from one of these strains here, put it into another background, and then you see that it's perfectly able to reduce the size of the plant and induce necrosis. So Wang Chengzhu, who has his own lab at Chinese Agriculture University now, then ran a GWAS, is a really cool GWAS, I think, because we only took accessions that had this specific allele, so fixed the background, and then asked, are there other common modifiers? And so you see two very nice hits. These are the 5 chromosomes of Australiana, 2 very nice hits. And Wang Sheng focused on MHA1, modifier of hyperactive ACD6, which encodes a small protein. And so this is the suppressor allele, this is the standard allele, there's a single amino acid change, and this amino acid change is a residue that is conserved in these other species, Lyrata and Rubella and Brassica napus. There is a close homologue in the genome, MHA1-like or MHA1L, and it actually has a threonine here, which is similar to the serine here in the Ty0 allele. So I'm skipping over a lot of the evidence. So the MHA1L seems to be the primary mediator of pathogen response in the standard strain of Arabidopsis thaliana. So we focus the mechanistic work on MH1L, which we did together with Uwe Ludewig's and Chen-Yang He's lab. And this is an experiment in mammalian cells and HEC293 cells and human cells. And this is just to show when you express ACD6, you get calcium influx. If you have this gain of function allele, you get even more calcium influx. So, and we also have evidence from Xenopus oocytes. So ACD6, if you put it into mammalian cells, so no other plant proteins present, it can stimulate influx of calcium. And that's further stimulated if you add MH1L as shown here. So MH1L can further stimulate this calcium influx. And then this is just genetic data to show that when you reduce external calcium, the phenotype of the ACD6 gain-of-function allele is largely suppressed. So this is the summary of considerable amount of biochemical and cell biology work, that you have these 2 versions of ACD6, the standard version that we find in the COL0 strain, and then this other version, EST-1. This is monomeric in the plasma membrane. It has these N-terminal anchorin repeats, intracellular anchorin repeat. So in the resting state, because it's a monomer, nothing happens. When a pathogen is detected, for example, through perception of FLAG22 and action of FLS2 and BAC1 co-receptors, MH1L is actually transcriptionally activated, the protein is made, and it acts as a ligand to enable dimerization of ACD6 protein through the anchorin, through the anchorin repeats, and now you have calcium influx. And calcium, I should mention, is a well-known secondary messenger in immune response. We can just overexpress MH1L in a 35S MH1L with a 35S MH1 construct. You force anchorin repeat dimerization, and you get massive influx and massive autoimmunity. Now, ACD6 EST-1 has this other version here, which spontaneously dimerizes, so spontaneously forms forms channels in the plasma membrane and therefore gives you spontaneously calcium influx, and you have autoimmunity in the absence of a ligand. And there's very interesting genetics. So MHA1L actually does not further increase this. MHA1L only interacts with the ankyrin repeats of the standard version. It does not interact with the ankyrin repeats of EST-1. And then MHA1, which is this paralog, has a special little TY0, where here also the small protein interacts with the ankyrin repeats, but rather than enabling dimerization, it actually prevents dimerization and thereby blocks autoimmunity, because now ACD6 EST is present as a monomer again. And this is just summarized here, this allele and paralog specific interaction. So ACD6, we have these two different alleles, and they interact with either MHA1L or this paralog MHA1, but only this TY0 allele, which we think it actually is basically moonlighting allele, if you will, that there is the standard version that probably interacts with an ACD6 homolog. So again, this is a very nice example of how the action of these different alleles completely depends on genetic background. And having the system very much, suggests that this has all been selected on. There was a very nice paper from my colleague Zach Lipman recently, where they looked at this actually on a genome-wide level in the genus Solanum, to show that paralogs act as contingencies in crop engineering, and obviously not just in crop engineering, they're contingencies in evolution in general, as I showed you with this example. All right. So I could stop here, but I still have a few minutes, and that's why I have to tell you about the somewhat annoying result that comes from the work of, first Talia Karasov and then Derek Lindberg. So what I just talked to you about ACD6, this is all a story from the lab, and it sounds really beautiful, all makes sense. We have these allele and paralog specific interaction, seems to be the outcome of evolution. But we wanted to know what happens in the wild. So almost 10 years ago, we said, okay, let's go out and collect microbes of Arabidopsis all over Europe to get an idea what are really the microbes that you have on Arabidopsis. So this is work led by Talia Karasov, who is now a professor at the University of Utah, and Rebecca Schwab, who's still at the Max Planck Institute. And several teams went all over Europe during a few months. Also, quite a few people went to Spain here. Again, I wonder why Spain was so popular in February. And so collected Arabidopsis populations from the Atlantic coast all the way to the Caucasus, from the Mediterranean here to the Baltic Sea. And we looked at the microbes in and on Arabidopsis leaves, and were quite surprised to find that there were two clear clusters, a southern cluster and a northern cluster of microbiome composition. They are similar at the class level. You don't see differences here between the two clusters, and the difference is all in individual strains. So here's one example. The most common microbes in Arabidopsis leaves, in and on Arabidopsis leaves, are Sphingomyces and Pseudomonas. Here's a Sphingomyces phylotype, is essentially strain where you see is a very nice latitudinal decline, less common in the south, more common in the north, and here's another one that goes the opposite way. So we then ask what explains having these different microbiome types, and it turns out that this is best explained by drought. So these are the different explanatory variables we looked at, and by far the best was Parma Drought Severity Index, which you will remember from one of my very first slides here. So drought apparently affects microbiome composition. Some of this is probably a direct effect of drought, but because we also know the genetics of all these strains, we can disentangle genetics and non-genetics, and we find that genetics is, that some of this is mediated by plant genetics. And that, of course, was very nicely when it was our earlier work, the work by Moy on drought survival, some of which I'd shown you before. So because we know it's genetic, we can then ask what are actually, are there individual genes that actually are responsible for this? So looking at all the immune genes in the genome, we find that the gene that is most highly associated with microbiome differences is ACD6. So I could stop now, this sounds pretty good, right? So ACD6, I'd shown you they had different alleles, and that seems to make a difference to the plant, and now is the evidence ACD6 really does something in the wild. To be fair and honest, I also have to discuss the recent work of Derek Lundberg, who is now a group leader at SLU in Uppsala. And Derek said, okay, this is all fine, but this is all association, this is indirect, it's correlative, let's do one final experiment. Let's grow plants with and without these ACD6 alleles in the field and ask what happens in the field, and that's what he did, unfortunately. So, in the greenhouse, everything is good, so you have this ACD6 allele, you are small, you have necrosis. But then you grow the plants in the field, either in Tübingen or in Zurich, and this, by the way, was, as far as I know, the first field experiments with genome-edited plants in Europe. We're very lucky that we had access to a secure facility belonging to ETH. You see these differences disappear when you grow plants outside. And obviously outside, you see here some of the plants from the experiment in Tübingen. Conditions are a little bit different from the greenhouse. So these plants are covered by snow in the middle of winter. So phenotypic differences disappear at the RNA-seq level. This is in the greenhouse. You see can nicely distinguish strains that differ in their ACD6 genotype, but you go to the field, these differences completely disappear in two different genetic backgrounds. So phenotype disappears, the expression differences disappear, and we thought, okay, so let's give it one more shot. We are really interested in the microbiome, and maybe there are no phenotypic differences, but maybe early on the microbiome is assembled and that the microbiome is a microbiome difference. So we looked at the microbiome, and long story short, when you look at the clustering here, see microbiome looks different in the field than in the greenhouse, and it's also different in different years, and either in the greenhouse or the field. But when you look at genotype, you see genotype makes no difference at all. And these field plants, actually here, they are much more similar. Their most similar samples are not the same genotypes grown in the greenhouse, but actually random wild plants that were collected also in Tübingen. So these blue one here, wild plants of completely different genotypes. So here, really, the environment is much more important than anything else. I wish I would have a way to conclude this story where there is all of a sudden a turn to the better or whatever, but no, there's not. So Derek also looked at fitness, and where is a clear difference in fitness in the greenhouse, the plants with the Columbia allele, the Col0 allele make twice as many seeds. When you go to the field, we don't see any differences anymore. So we are a little bit left here shrugging our shoulders, and we might want to conclude there are no fitness consequences in the field. Well, there we have to pause and think about a little bit what we have actually done. So we have grown plants in 2 seasons, in 2 different field conditions. So does that really mean that there is no difference in the field? I think what it means is there's no difference in our field experiments. And this is a principal problem when doing field experiments, that you choose some quite arbitrary environment. If you see something, is fine, but if you don't see anything, it actually doesn't mean a whole lot. And we recently published a commentary on this with Derek and Talia and my colleagues Joy and Fabrice. When is the right time to work in the lab? When is the right time to work in the field? And what do results from one mean versus the other? So what I want to leave you with, all of you know this first quote, one of the most famous quotes in biology, perhaps the most famous quote in biology, that nothing in biology makes sense except in the light of evolution. This was somewhat modified, this beautiful book by Rosemary and Peter Grant on speciation, where they concluded the book with a statement that nothing in evolution makes sense except in the light of ecology. And what I want to leave you with, that really nothing in genetics makes sense except in the light of ecology. So it's important what we do in the lab, and we can construct really beautiful stories, but in the end, one should endeavor to go out in nature and try to test at least a few of the things that you found in the lab, also in wild environment. And I just leave you with some of the images of what it looks like when you sample Arabidopsis thaliana in the wild, and I thank you for your attention. I'll be happy to answer any questions you might have. Thank you.
Thank you very much.
And I prefer if students and postdocs ask questions first, and I have no problem standing here silently for a minute or 2 or 5 until a student or postdoc takes mercy on the audience and asks a question.
So who is the first student who has questions, or a postdoc? Nobody?
No. We just wait. We just wait. So we just wait. So it's perfectly fine. So I'm used to this. So we just stand here in silence. So if somebody, until somebody has, no. We'll just wait. So I'm sure somebody will come up with a question.
People.
Yeah. No.
So now
we just wait. We just wait.
Yeah.
So we just wait here. So.
Sit down. So
They're cookies, so you know. Yeah. So there's a reward.
In the coffee break, probably. Yeah, there's one student.
Oh, right.
Let's give him an applause. Okay.
Excellent.
Camera.
Excellent.
Thank you for all. The first thing, I would like to know what conditions do you think that could be the best for do this kind of experiment? As you mentioned before, it couldn't be the best conditions, as you didn't see anything about the gene in these conditions.
Yes. So thank you for that question. That's what I think about all the time, and it's sort of like, is your cup half full or half empty? So I think it's actually interesting to go back and think about, if you do biochemical experiments in the lab, okay? Do biochemical experiments in the lab that often the way it works, you do your experiment 100 times and it doesn't work the first 98 times, then it works once, and then Pablo comes and says, repeat it maybe once. And then if you're lucky, it works again, and that has worked 2 out of 100 times. And you take that actually for, so it worked. And so I think it's really similar with field experiments, that to know what matters in the field, you would have to do this experiment over many years in many different conditions. And that's actually what plant breeders do. So plant breeders, for example, only, so if somebody wants to sell a new variety of seeds, they will only do this after they have grown their new variety in many different fields in over, at least a few years or so. So the answer would be you have to try out a large variety of places. You have to grow the plants at different times, and you have to do it for many years. And that's also what makes it so difficult. You can't do that for every test. So you have to be really smart about what you wanna test. But there's a beautiful book by my colleague Annalisa Leine, who has thought about this much more deeply than I have, on these sort of interactions. And the conclusion, more or less, that Annalisa comes to in this book is that it's almost impossible to show that these sort of interactions between microbes and their host, what the ecological relevance is, because it's so highly context dependent, and that you basically can never do enough experiment or collect enough samples to show this. So in the end, I hope that the experiments in the lab do mean something, that these alleles are not just there by chance, but you probably have to look at many different situations, a lot of different type of evidence to answer this sort of question. All right, at least one more question from one of the junior researchers.
Hello, thank you very much for your presentation. I don't know if it's a question or a commentary. For me, in the evolution now, try to survive in different environment with all these changes. Now with your presentation, it's like, okay, 2 plants from 2 different countries, that will be the maximum of genetic variety. Now is bad for the plant, no? So I don't know how could you explain that, because the genetic variation is good for evolution, but somehow sometimes good, sometimes bad. I don't know if.
Yeah, so this is just another example of these trade-offs. That genetic variation on the one hand is you need genetic variation so that evolution can proceed and that you can also survive in different environments. But then genetic variation can also be harmful. So another really cool human genetics paper that I love is a paper from Kari Stefansson from Decode Genetics. So it's about Icelandic population, and so in Iceland they have really good records of everybody, how they got married and children and whatnot. And so they looked at what genetic distance gives actually the highest fitness. Okay, and the fitness they measured in also just measured in the number of children. And so it turns out in Iceland, it's the best if you marry your 3rd cousin. So, if you which is a little bit of an uncomfortable truth, if you will, but. So that there is basically an optimum. If you're too close, then obviously, the likelihood that some genetic disease got exposed is really high. But if you are too far away, then there is a genetic mismatch. So this is one paper, whether what applies in Iceland applies elsewhere, who knows? But it sort of intuitively makes sense that there is some, genetic optimum for crossing. So, but Arabidopsis thayana is actually it's a really cool species to study because we have very different types of genetic diversity. So we work a lot in south of Germany, and then we work with Fabrice Roux a lot in the southwest of France. And so in Germany, if you go into a single population, everybody is almost the same, but then you just go 100 meters away, they're completely different. And in France, it's the opposite, where in a single population, everybody is completely different, but then you go 100 meters away, you wouldn't really know that you're 100 meters away because they're very similar to this other population. So Arabidopsis is actually a really nice organism to study these kind of questions. There was another question.
See, so mine maybe is a silly question. So I've never done experiment collecting samples, but as there is so much phenotypic diversity, how do you know, or is it the case that sometimes you get a sample of something that is not actually Arabidopsis thaliana, or what is the characteristics that you follow for knowing, or you sequence, or?
Yeah, so no, that's a very good question. So first of all, we train people in the lab, and it's really quite amazing once you have a trained eye how easily you can recognize them. And so one of the most astonishing observations in that direction is I was recently lucky to visit the Kew Botanical Gardens. So Rafael Gutaka, who was a student with a postdoc with Hernan Bobano, and I asked him to show me the Arabidopsis collection. And so we went, it's a whole books of Arabidopsis collection, and they had samples from the tops of these African mountains. And so there, because of the environment, they are all dwarf. They are all GA, they are impaired in GA production, actually, the plants. They are mutants. And they look really, really different. So they are, so they don't have a main inflorescence, they just have many shoots. And so as a layperson, you look at this and you say, this is not Arabidopsis thaliana, but whoever collected these plants actually realized they are Arabidopsis. We also, of course, mostly sequence, then we also know that, you know, it's what we want. Yeah, our false positive rate is just a couple of percent, so it's both training and genetics. Share one more thing with you. With Joy and Fabrice, Luke Henry as first author, we recently submitted a paper for, I think it was molecular ecology or something. And there we described that we were looking at Arabidopsis and these companion plants, and we used MET-case sequencing to identify these companion species. And we collected them vegetatively, so it's a little bit challenging to know what they are. But one of the reviewers said, it is depressing to see the modern state of ecology where the people who go out sampling have to sequence the plants to know what they are. So we said, well, sorry, that's what we did. So.
First of all, thank you so much for your amazing talk. One of the messages that I got through the talk was, well, the question maybe could be very naive, I don't know. But is that each species has a different tolerance to the amount of NLR proteins. So what could be the determinants for these different tolerance through the species?
Yeah, so I assume you mean what would be the molecular biochemical determinant. So in the ecological thing, you know, I would say it has to do with, you know, what is the chance that they encounter, you know, whatever they encounter. This also speaks to, are they generalists or specialists? That's something that I didn't, you know, touch upon. So basically, these ACD6 plants, we can think of them, they are generalists. They resist everybody, but it comes at a cost. And then specialists are the ones that, you know, only when there is attack, they do this. So I think at the biochemical level, it most likely has to do with chaperones that help assemble these resistosomes, how easily they assemble. That's how I think about this. It's a little bit difficult to do, you know, genetics between papaya and wheat. This is in the end what one should do. But, you know, if one could somehow figure out, you know, say, put the papaya genome into wheat cytoplasm and see what happens, that would be the right experiment. So yeah, no, but it's something that I totally think about all the time, and I would love to have the answer to. So, but I haven't been able to come up with a clever experiment to test this.
Yeah, there's one very young student here.
I'm still a postdoc. Yeah, I wanted to ask about the last part, the field experiment. Can it happen that you have a trade-off, like this growth immunity trade-off? Do you have additional trade-offs that you are not aware of them? Like this happened to us even in growth chambers. Like we have single mutants that here we see clear phenotypes. We do it in a collaborator lab. We don't see the phenotype anymore. We just think there's something we are not taking in account that is also important for the phenotype. You
put your finger actually on what is the biggest issue with field experiments, right? So, you know, we think what matters in the field, but how do we really know that this is what is really important to the plant, right? So we do the, you know, drought versus non-drought, or the pathogens versus non-pathogens, and it's just really, really difficult to know. So we recently did some super simple experiment where we just collected lots of plants from outside, and we just looked at their transcriptomes, and the idea was to ask, can we classify these transcriptomes, that some plants are more in the drought stress mode and other plants are more in the immunity mode and whatnot. And what we find is that it's basically this smear of states that so in the lab, you know, we torture the plant this way, we torture the plant that way and whatnot. And it's very clear, but when you go outside, apparently that doesn't exist. So the plants are always in some kind of in-between mode. So when you read the literature, the ecological literature talks mostly about the two axes that I described here, or maybe three axes. So there's drought, there's pathogen, and then there is competition from neighbors, shade avoidance. So those are in the ecological literature. well, maybe pollinators, so Arabidopsis doesn't need pollinators. But those are sort of the main axes. But in the end, is that what really matters? Who knows? And so maybe one more thing to share. I have a colleague in Tübingen, Katja Thielberger. She is an ecophysiologist. And Katja once told me that ecophysiologists actually have a saying: if an experiment is nice, don't do it twice. And so what she explained to me, what is meant with this, if you're a good naturalist, okay, so you walk around outside and you observe things, you see certain patterns, and then in your mind you formulate a hypothesis, as we do as scientists, right? But then you sort of already know the answer. Then you do a formal test, you see, okay, I take this plant there and there, I test this, statistical significance and whatnot, and I publish this paper that says evidence for blah, blah, blah. And what she meant was, yeah, you picked that example that, confirms your hypothesis, but good luck if you want to, show this with some other system, So I have not come to any good answer how we, figure out what plants really need and want. So if any of you have an answer, please tell me.
Yeah, next student is Pablo.
Student of life.
Exactly. So let's start with a statement. Nothing makes sense in genetics beyond the
except in the light of ecology,
of epigenetics.
Mm-hmm.
So
okay. Except in the light of epigenetics. Right. Very good. Okay.
So the question is: so you collect the samples and you end up multiplying those seeds in the lab. So have you seen a diluting effect on the phenotypic variation with the generations when you multiply the seeds on the lab, or in the other way around, when you take the seeds from this field experiment that are showing no effect, and you go back to the lab, you still see the foundational effect, or you lose it?
Yeah. So right now, this is certainly a question that many of my, or not many, but a number of my colleagues are super interested in this question of phenotypic variance. So where they look at the CV, the coefficient of variation, and so it's the broad umbrella is the evolvability of evolvability. So can you actually be in a state where you change in more quickly and less quickly? And we have not looked. So in Arabidopsis thaliana, I don't think. Well, we certainly haven't looked at, and I'm trying to recall whether anybody has looked at this. I think there are few, if any, papers that have looked at phenotypic variance where you're genetically identical. So I think that hasn't really been done. So we certainly haven't looked at that, whether phenotypic variance changes. I mean, from first principles, for sure, there are maternal effects. So you would, if these maternal effects should go away as you propagate plants in the lab, because they are more and more homogeneous. So for sure, without having done the experiment, I can guarantee you that first generation plants are going to be much more variable than 10th generation plants. But whether beyond the next few generations this changes, I don't know. You may remember we did this experiments with Anja Wibobo and paper Marcos Gutierrez, where we drought stressed plants, and we showed that there was an effect in the immediate offspring, but then 2 generations later it already disappeared. So. But that seems to be, this epigenetic memory seems to be also highly variable between different species of plants, just from I see from the literature.
So since I have a microphone, I do another one.
All
so
right.
This is more philosophical. So you were working for quite a long time of sequencing plant with 1001 genome and short reads and stuff. What did you learn now that you are doing like long read genomes? So much of difference is quite cool stuff that you can find now that you miss it like 15 years ago, or
so first of all, so much of what we do is based on what people in the human genomics field do, and it's really quite amusing to see this. So the human genetics field, it's I wouldn't quite say they are charlatans, but they are really good in misleading advertisement. So initially, when people like Francis Collins went to, went to the taxpayer, to the, president and so on, they said, it's going to cost a lot of money, $3 billion, to sequence the human genome, but that's going to be the answer. Well, now, they sequence human genome, and they say, oh, we forgot. Humans are actually not identical. There's not just one human genome. We have to look at SNPs. So give us money to do SNPs with, microarrays, and that costs I don't know how many hundreds of millions. And we're like, oh, that was pretty good, but actually we need higher density of SNPs. So give us, a few 100 million or billions or whatever to do short read sequencing. And now it's, we need even more to do all this long read sequencing. And so what we have actually learned when it comes to discovering genetic variation, short read sequencing is 80 to 90% of what is there. So all these papers, they're always like, so long read sequencing, it gives you all this more information. Yeah, it gives you more information, but the actual gain in information compared to the cost is pretty minimal. So you get actually a lot from short read sequencing would be the answer. It's a lot easier with long read sequencing in some ways, but it's also a lot more difficult with long read sequencing because this may be a surprise to many here as it was to me. So I did not appreciate until recently. So we were some of the first who did genome graphs. We did this already, over 15 years ago. One of the first papers on genome graphs was from my lab, Corbinian Schneeberger and Jörg Hagmann. And then we've been working on genome graphs since then. And it was really difficult. And I got really frustrated. There were a number of graduate students really sort of dissertations that didn't go anywhere, and I was like, what's wrong here? Why are we not making any progress? And it turns out that whole genome alignment is a profoundly unsolved problem, and it's probably an unsolvable problem. So to interpret long read genomes, you actually have to align genomes to each other, and that there is not a simple answer. There isn't no real answer to this. So it really depends on what you want to see and where you are in the genome. Whereas to align short reads to a reference genome, that's a really straightforward process that we understand. But to align two different genomes, even if they are really similar to each other, is an unsolved computational problem. And anybody who wants to talk to me about why that is, I can talk to you about this. But it's the summary is, whole genome alignment, as you call it whole genome, you have to use one set of parameters to align two genomes to each other. But in some places, the genomes are very similar. In other places, they're not very similar. So you actually have to do it in a dynamic way. But you can't do it in a dynamic way because it's a whole genome process. The bottom line is, long read genomes, it's a little bit of, we're doing a lot of long read genomes. We have, over 500 long read genomes, but you don't learn quite as much as what, all the papers might want you make believe. Sorry.
Hi, Detlef. Beautiful talk.
Thank you.
It's a quick question. How much of what you learn in Arabidopsis, which is auto-pollinated plant, can be translated when you talk populations that they have to be cross-pollinated? I mean, how?
Yeah, so this is actually one of the beautiful things about Arabidopsis. It's when you go to the field, most of the individuals that you pick are highly inbred. Actually, a famous population geneticist, Chuck Langley from UC Davis, one of the early human geneticists, told me 25 years ago that I was working on the wrong organism, because he said basically if you inbreed, that's the path to extinction. Why would you even work on Arabidopsis? Because it's a species that for sure is going to die out. But it turns out that Arabidopsis crosses out around every 10th generation, and if you look at the properties of the genome in terms of linkage disequilibrium, the genome looks practically like a human genome. And so humans actually inbreed quite a lot. So especially if you go to the Middle East, for example, it's not unusual. There are many individuals, 70% of their genome are actually homozygous or autozygous, as human geneticists will call it. So Arabidopsis outcrosses often enough that it's you do learn from outcrosses, but it has, of course, overall less genetic diversity than outcrosses have. And one of the things that I've always been interested in, this genetic incompatibility. What does it look like in outcrosses? And we've been trying to answer this together with Kirsten Bomleys by looking at Arabidopsis arenosa, which is an outcrossing species, and don't have an answer yet.
Okay, any other questions?
Hello. In my former lab with Peter Broderson, as you may know, we were quite obsessed with autoimmunity, such that every time we saw a mutant that was a little bit smaller or whatever, the first thing we thought was autoimmunity, and very often we were right. And then we were working with some mutants that sometimes show autoimmunity, or what we thought it was autoimmunity, sometimes not. We wanted to push it, and then it was right after I had some holidays in Patagonia, and then I was really observing the plants there and the temperature cycles, and then I thought, of course, in Denmark, the temperature variation is between, I don't know, +1, -1 the whole winter, and so is in the Percival. So maybe it's not about cold or hot temperatures, but about the daily variation and the randomness of it. So we designed what we call the Patagonia experiment. So we programmed a chamber with very sharp temperature shifts, and that it wouldn't repeat. So every day would be different. But we had the program, and then we just got random results. So not only because the program was random, but we could repeat it. So every time we ran that random program, we got completely different phenotypes. And we never had an explanation for that. And I wonder if you have some insights on why, when you repeat a random program, the phenotypes of the plants are so different every time.
Yeah, so thank you for asking that. So I think the simple answer is that this is, it's the butterfly phenomenon, right? So if that a butterfly in the Amazon can lead to a hurricane in North America, where you repeat the experiment, but really there are very small differences in the experiment that then lead to these very different outcomes. So the stochasticity effect, I think, is well known in science. So I think this would be the simple explanation. But I really love that you did this experiment. So we know for the ACD6, we already know that it's not the temperature per se that matters, but it's actually the difference between night and day. And this is something that Derek, together with another postdoc, Rosa Leitinen, are now working on. So I think this experiment is really cool. So it was the right way to do the experiment. The way you did the experiment was a complete randomness, but then also you made it particularly challenging for yourself, I think. And then also one more thing is that, for work from Shenyang He, we now know that some autoimmunity mutants show autoimmunity only in the presence of microbes, and others do it without microbes. And the ones that we work on, they show no microbes are needed for autoimmunity. But I love it that you did this with completely random cycles. That's really cool.
To me, what was remarkable is that every time all the plants look, not all the plants, especially the wild-type control, all the plants look the same. So it was not that in some individual something got triggered and not in another. But then when we took pictures, and then we repeated the whole cycle, same seed batch, same hands, different phenotype in all the plants. And that's completely crazy. So it must be, I mean, that's sort of crazy to think that, it must be very small differences, but then that the differences are actually fixed in the plants and the soil that you use and everything. So that is really insane. So, but it could be, for example, if it's a phenotype that is microbe dependent, that it could be, you use different soil and very small difference in the microbes makes a difference to the plants. But that's totally fascinating. I would totally work on something like that. So.
Well, thank you very much to all of you, especially to students, young students, and now we can give another applause to Detlef. Thank you.